Hi, welcome to this session of fetal cardiology. We will be discussing today a bit of fetal cardiac embryology, fetal circulation, and transition to postnatal life. So, fetal cardiac development is a continuum from the embryo through the fetus to postnatal life. During the embryonic stage of the fetal heart, the growth of the heart is predominantly by the process of hyperplasia, where cardiomyocytes increase in number. This continues through fetal life until postnatally, the heart then starts to grow. So each cardiomyocyte then hypertrophies and grows in size. At each stage of development, there are a variety of intrauterine and extrauterine factors that have an impact on the development of the fetal heart. So up to 3 weeks of embryogenesis, oxygen passively diffuses through the placenta, and this is sufficient to sustain the embryo. But beyond 3 weeks, this passive diffusion is inadequate for further growth. So around this stage, around day 22, is when the first fetal heartbeat is noted in the primitive heart tube. Now, from that stage, the heart develops pretty rapidly, so that at the end of 7 weeks, there is actually a proper 4-chambered heart. So if we look at the embryological development of the heart, the origin of the cardiac cells actually arise from the mesoderm and the ectoderm. So both the cardiac mesoderm and the ectoderm provide progenitor cells to various portions or parts of the heart. So the first thing that we notice during cardiac development is the formation of what's called a first heart field. So these are the very first cells that commit themselves to forming the heart. Then we have the second heart field. These are the second wave of pluripotent cells that again commit themselves to forming the fetal heart. So this is the first step in cardiac development. Now, by the time the embryo is about 22 days of age, what happens is this first heart field forms a tube, okay, which is called the primitive heart tube. So you can see that here, the first heart field, you can see the cells belonging to the first heart field are colored in a orangey yellow color, and that forms the primitive heart tube. The second heart field seems to be committed to forming the outflow tracts. What's labeled OFT here is the outflow tracts. So the primitive heart tube initially starts pulsating. It starts to have peristaltic movements like pulsations at about 22 days of age. Now, from 22 days onwards, what happens is that this primitive heart tube loops in a rightward fashion. Now, actually, the first heart field is anterior, the second heart field is posterior, but the looping of the primitive heart tube, what it does is it brings the posterior portion anteriorly. So when the primitive heart tube loops to the right, the cells belonging to the second heart field actually come across anteriorly. And the second heart field gives rise to the anterior right ventricle and the outflow tracts, the origin of the outflow tracts. The primary or the primitive heart field is posterior, and it gives rise to the left ventricle and portions of the atria. Okay, so there is a complex looping process that happens during embryological development where the primitive heart tube loops in a rightward fashion. It brings the posterior second heart field anteriorly, and that forms the right ventricle and the origin of the outflow tracts. The primitive heart field itself then becomes more posteriorly placed, and that becomes the left ventricle and the portions of the atria as well. So you can see that in the last image, the left ventricle and the atria are colored in the same yellow as the primitive heart field. So these are actually derived from the primitive heart tube and the first heart field. The portion in blue, which is the right ventricle and the origin of the outflow tracts, that is derived from the second heart field, which forms the outflow tracts in the anterior right ventricle. Then, of course, you have additional cells which are derived from the ectoderm, which are the cardiac neural crest cells. These are in green. These cardiac neural crest cells actually form the great arteries, they form the valves, and they also form the coronary arteries, etc. Okay, so they actually are responsible for outflow tract septation. So these neural crest cells are very important in the proper development of the outflow tracts. So there are, in this embryological development of the heart, there are a variety of transcription factors and genes that are intricately involved with the development of the heart at each stage. There are a couple of transcription factors, which are the mesodermal factors. These are the original factors that are seen right at the beginning of the development of the heart. So you have the mesoderm posterior 1 and 2 factors. These are transcription factors that are actually involved very early on when the first and second heart fields are formed. So they actually are responsible for the formation of a reservoir of cardiac myocytes, which then increase in number by hyperplasia. Then subsequently, you have T-box transcription factors, which are TBX2, 3, 5, 20, etc. And these T-box transcription factors are responsible for cardiac chamber morphogenesis. In addition, there are other transcription factors, GATA4, NXX25. These are all involved in chamber myocardium specification. So any abnormality in these transcription factors will lead to a maldevelopment of the cardiac chambers. So we've gone through all of this. The neural crest cells, like I told you, are responsible for the outflow tracts, and these are responsible for outflow tract septation. Notch1 and TBX1, these are some deletions that can cause disturbances in outflow tract formation, and these are associated, these transcription factors are associated, deletions are associated with DiGeorge syndrome, for example. DiGeorge syndrome, as you're probably aware, mainly related to outflow tract abnormalities. So now that we have a broad idea of the embryology of the heart, let's move on to fetal circulation and physiology. Now, this image illustrates our journey from studying fetal hearts in sheep to now studying human fetuses by means of cardiac MRI. So our initial knowledge of fetal circulation was derived from the lamb fetus, and several lambs had to undergo experiments in order for us to learn about fetal circulation, sadly. However, nowadays, we are able to use cardiac MRI to determine many aspects of fetal circulation, and that's really a great step forward, particularly for the poor lamb. Okay, so what are the specific peculiarities or features of fetal circulation? So, like we said earlier, in early life, oxygen is able to, transfer to the embryo just through plain diffusion. But subsequently, this diffusion is inadequate to sustain the embryo. And during later fetal life, what we need is a proper fetal circulation. So how is this fetal circulation established? So basically, the placenta derives maternal blood, and through the placental plexus of villi, blood, oxygen, nutrients all of that is transferred along to the fetal circulation through the umbilical vein. Okay, so this umbilical vein then enters the fetus and becomes the main source of oxygen and nutrient supply to the fetus. The umbilical vein, as you can see illustrated in this beautiful diagram, actually supplies a part of the liver, the left lobe of the liver. Then it gives off a vessel called the ductus venosus. The ductus venosus is the main vessel through which the fetal heart and fetal circulation get oxygen and nutrient-rich blood from the mother, from the placenta. So this ductus venosus is the connection between the umbilical vein and fetal circulation. So the ductus venosus goes and joins the IVC, enters the right atrium, and is channeled through the foramen ovale into the left atrium. The umbilical vein also continues onward to partly supply the right lobe of the liver. So the umbilical vein gives off some branches to the left lobe of the liver, then gives off the ductus venosus, and then also somewhat supplies the right lobe of the liver. So at least 50% of the umbilical venous flow passes through the ductus venosus and contributes to fetal circulation. This percentage of flow is highly variable in fetuses. There is a wide range on fetal MR when this has been studied. There is a wide range between 20% and 90%. But on an average, 50% of umbilical venous flow goes through the ductus venosus, enters the right atrium, but is channeled through the foramen ovale preferentially into the left atrium. And we will have a look at the mechanisms of this. So why is this ductus venosus flow preferentially channeled through the foramen ovale into the left atrium? There are several theories. One of them is that the eustachian valve helps to channel the blood. The other is the difference in velocity between the IVC blood and the ductus venosus blood, with the ductus venosus having a much higher velocity and therefore jetting across the right atrium through the foramen ovale into the left atrium. Now, this umbilical venous blood, which is going in through the ductus venosus, has a much higher saturation than any of the other, blood flows that come into the heart. And that is why it is important that it goes to the left side of the heart. So the most oxygenated blood is actually in the ductus venosus. So here, this is an illustration beautifully by an expert who has done a lot of experiments in fetal sheep, Rudolph, who has published a book and published several articles on fetal cardiac circulation in sheep. And this is one of his very nice illustrations where you see the oxygen saturation in the various chambers and vessels of the heart. So here you have the IVC, the ductus venosus, which has the highest saturation of 70%. And the ductus venosus blood preferentially streams across the foramen ovale into the left atrium. The SVC blood and the actual IVC blood, not the ductus venosus blood, but the IVC blood, then go along across the tricuspid valve into the pulmonary artery. And this is much more desaturated than the ductus venosus blood. The ductus venosus blood has a saturation of around 70%. That higher concentration blood, along with the pulmonary venous blood, then goes through the left ventricle and into the ascending aorta, supplying the coronaries and the carotids. Whereas the lower oxygenated blood from the IVC and the SVC pass through the tricuspid valve, through the pulmonary artery, through the PDA, the ductus arteriosus, not the PDA, but the ductus arteriosus in the fetus, and down into the descending thoracic aorta, supplying the abdominal organs. So the more oxygenated blood is in the ductus venosus, it streams through the foramen ovale, into the left side of the heart, aorta, supplying the coronaries and the carotids predominantly. The less oxygenated blood from the IVC and the SVC passes through the tricuspid valve into the right ventricle, the pulmonary artery, the ductus arteriosus, and the descending aorta. So now we have understood one of the peculiarities of fetal circulation, which is how the ductus venosus helps to channel more oxygenated blood into the left side of the heart to supply the brain and critical organs. Then we move on to the actual cardiac output in a fetal heart. So generally, we all know that the right ventricle is the more dominant ventricle in a fetal heart. So if we look at the output of the right ventricle versus the left ventricle, right ventricular output is more than the left ventricular output, which is not the case in postnatal life. Postnatal life, the right ventricular and left ventricular outputs are equal. But here in fetal life, right ventricular output is greater than left ventricular output. So the ratio in the fetal, the human fetus is about 1.3:1. In the sheep or the lamb fetus, it's actually 2:1. So it's much higher, that ratio. So fetal MRI has once again helped us, to see that the human fetus is slightly different in this aspect. Now we see that during fetal life, there is only very limited pulmonary blood flow. Now in fetal sheep, in early trimesters, there is only 4% of the cardiac output going to the lungs. And at later trimesters, near term, about 8% of the cardiac output goes to the lungs. In the human fetus, at 20 weeks gestation, 13% of cardiac output goes to the lungs. And closer to term, at about 30 weeks, 25% of the cardiac output goes to the lungs. So this is again new knowledge, because originally it was thought that the human fetal heart, which was relatively similar to the sheep fetal heart, we were under the impression that pulmonary blood flow was extremely limited throughout pregnancy. But what we have learned is that in the later stages of pregnancy, nearly a quarter of the cardiac output goes to the lungs. Again, foramen ovale flow, in earlier pregnancy, there is greater flow through the foramen ovale. This is a right to left flow, and this constitutes nearly 35% of cardiac output at 20 weeks gestation. But what happens in later gestation is that there is greater pulmonary blood flow, greater pulmonary venous return, higher pressure on the left atrium, and therefore the right to left flow across the foramen ovale almost halves. Okay, so this is another important thing to remember. Again, the flow through the umbilical vein is much higher in earlier gestation and lower in later gestation. So this in the fetal lamb heart, there is not that much of a change throughout pregnancy. But in the human fetus, there is a change. There is a decrease in the umbilical venous flow in later gestation. So near term, it's only 120 mils per minute per kg, whereas in early gestation, it's about 140 to 150 mils per minute per kg. Now, how does the fetus compensate for this reduction in flow? Is mainly through fetal hemoglobin. So when we look at the fetal hemoglobin levels, the hemoglobin concentration in early trimester, it's only about 10 grams per deciliter. And towards term, it goes up to as high as 16 grams per deciliter. So that's how it compensates for the reduction in the flow. So it increases the production of fetal hemoglobin, which has a greater affinity for oxygen and therefore maintains oxygen delivery to the organs. So what are the other peculiarities of the fetal heart? The fetal heart is believed to operate at the top of the Starling curve, which means that there is very little reserve capacity for the fetal heart. So this was originally published in one of the earlier publications, where they increased the preload and tried to see if that had an impact on the stroke volume and cardiac output. And they found there was no increase in the stroke volume or cardiac output by increasing the preload. However, further studies have shown that if the afterload is maintained, if the afterload does not go up, then preload increases can translate to cardiac output increases. But notwithstanding this data, the fetal heart does have much less reserve than the postnatal heart. Myocardial contractility is diminished because of the immature myocardium. There is less sympathetic innervation. There are fewer adrenergic receptors. All of this has an impact on the myocardial contractility. So if the cardiac output in a fetus has to be augmented, it has to come from mainly a heart rate increase rather than a stroke volume increase. But because of the reduced number of receptors, adrenergic receptors, even this heart rate increase is actually suboptimal. So in summary, this slide essentially translates to the fetal heart has only limited reserve, both from a contractility and an adaptive capacity point of view. Now, further, we look at the biochemical milieu, biochemical and hormonal milieu of the fetal circulation. In early gestation, a high pulmonary vascular resistance is maintained because of a high concentration of vasoconstrictors like endothelin-1 and prostaglandin F2 alpha. In later gestation, what happens is the vasodilatory substances like prostaglandin I2, which is otherwise known as prostacyclin, and prostaglandin E2, which is produced by the placenta, these predominate, therefore dropping the pulmonary vascular resistance, which, if you remember, the pulmonary blood flow goes up in the late trimester. So this is because of a drop in the pulmonary vascular resistance. So the biochemical milieu changes. Also, there is, we know that the fetus is constantly living in hypoxemia. So whatever blood that comes even from the mother is only 70% saturated. So even the umbilical venous blood is hypoxemic. So the fetus adapts itself to this hypoxemia by fetal hemoglobin and also running a higher heart rate to optimize or maximize oxygen delivery to the organs. So if we look overall at what are the differences between fetal and neonatal circulation, fetal circulation actually runs in parallel, whereas neonatal circulation runs in series. So there's the venous part in the neonate. There's a systemic venous blood, goes into the lungs, comes back as a pulmonary venous blood, goes out, cardiac output into the aorta, and then systemic venous blood. So it's in series. Whereas in the fetus, there is a lot of mixing at different levels, and the circulations are actually running in parallel. So whatever goes in through the pulmonary artery is not just going to the lungs, it's a small proportion going to the lungs, also going into systemic circulation. So the circulations are running in parallel. The site of gas exchange in the fetus, as you know, is the placenta. In the neonate is the lungs. Then the fetus has multiple shunts. That is the ductus venosus, which we've discussed, the foramen ovale, which we've also touched upon, and the ductus arteriosus, through which the pulmonary artery shunts blood into the descending aorta. In the postnatal life, all of these shunts usually become non-functional, either close up completely or start shunting left to right or become insignificant. Then again, in the fetus, the rate of perfusion is much higher than in the neonate. Heart rate is also much higher in the fetus than in the neonate. And in the fetus, the pulmonary vascular resistance is higher than systemic vascular resistance. In the neonate, systemic vascular resistance goes up because the placenta is removed from the circulation, and pulmonary vascular resistance drops because of various factors, including ventilation, oxygenation, vasodilatory substances, etc. Okay, so these are the differences between fetal and neonatal circulation. So how does the fetus prepare for transition? So basically, there is a change in the internal environment of the fetus where there is a downregulation of vasoconstrictive substances and an upregulation of vasodilatory substances. So the downregulation of cyclooxygenase. So there is a decrease in the production of a vasoconstrictor thromboxane A2. There's an upregulation of nitric oxide synthase. So there is an increase in the vasodilator nitric oxide. In addition, there is an increase in prostaglandin E2 and prostacyclin production. And all of this results in a drop in PVR. So in late gestation, drop in PVR, increased pulmonary blood flow. They are preparing for birth and postnatal life. Again, there is an increase in the cortisol and amine levels in the fetus through the later trimesters, getting ready for labor and postnatal life, and an increase in thyroxine as well. So all of these are for the fetus to transition into being a neonate. So what are all the postnatal changes that happen? So once a baby is born, the umbilical cord is clamped, and that means that the placenta, which is a low vascular resistance circuit, is removed from systemic vascular circulation. So the systemic vascular resistance goes up. Now, the baby also, because of chemoreceptor responses, starts breathing. Now, through labor, fluid in the lungs is already squeezed out, and surfactant has been produced by the pneumocytes inside the lung alveoli, trying to maintain the alveoli open. So once the chemoreceptors have been triggered, this makes the newborn take breaths, spontaneous breaths, and this results in an expansion of the alveoli along with intake of oxygen. So there is ventilation and oxygenation that happens, and both of these have a vasodilatory effect on the pulmonary vasculature. In addition, there is an increase in the level of nitric oxide and prostacyclin. All of these factors result in a drop in pulmonary vascular resistance. So this continues to drop and reaches normal levels by about 3 weeks of age. So when the pulmonary vascular resistance drops, what happens is there is an increase in the pulmonary blood flow because the resistance has dropped. Now the pulmonary artery wants to send blood to the lungs, not through the ductus to the body, because SVR has gone up, but PVR has gone down. So blood always takes the path of least resistance. So now the blood flows into the lungs, increase pulmonary blood flow, automatically, naturally increase pulmonary venous return back to the left atrium. Left atrial pressure goes up. Now there is a flap valve in the atrial septum. Flap valve shuts. Okay, so this flap valve originally in the fetus was shunting right to left. Ductus venosus flow was going from right to left through this flap valve. But now postnatally, there is no longer any ductus venosus. The umbilical vein flow doesn't exist anymore. There is no need for the flap valve, and increased pulmonary blood flow equals increased pulmonary venous return, high LA pressure. LA pressure becomes greater than RA pressure, flap valve shuts close. So no longer having a right to left shunt. So this happens more or less soon after birth, within a few hours. And the PFO or the foramen ovale can persist and be what is called a patent foramen ovale. And in a majority or quite a significant proportion of babies, this will close anatomically by about 3 months of age. 50% will close by 5 years of age. In a proportion of adults, in a third of adults, this foramen ovale can remain small but patent, but is usually of no significance, rarely of significance when there is paradoxical embolism and stroke, early onset stroke. But this is quite a rare phenomenon. In most adults, a PFO is an innocuous or benign finding. In fact, you and I might have a PFO. Okay, so this is as far as the foramen ovale is concerned. Then we move on to the ductus arteriosus constriction. So we know the placenta is removed from the equation with clamping the cord. The foramen ovale closes with a drop in PVR, increased pulmonary blood flow, increased pulmonary venous return, increased LA pressure. PFO closes, the foramen ovale closes. At least the right to left shunt ceases. There can be a small left to right shunt, and the foramen ovale can persist in a proportion of adults, but it's usually left to right. Now, next is the ductus arteriosus. This also constricts. Now, this constricts because of a high oxygen saturation in the blood, because of a high oxygen content in the blood, and also because of endothelin 1. Also, prostaglandin E2 used to keep the duct relaxed, and this prostaglandin E2 was produced by the placenta predominantly. Now there is no placenta, So prostaglandin E2 has pretty much disappeared. So all of these factors lead to constriction of the ductus arteriosus. Now, with the fall in the PVR, the duct, as it's closing, will start shunting left to right, because now SVR is higher, PVR is lower. Functional closure of the ductus usually occurs between 1 to 4 days. Usually, ductal flow ceases in normal term babies by about 72 hours, maybe 96 hours. Okay. But permanent closure, where there is endocardial proliferation and fibrosis, takes about 1 or 2 months to happen. But usually by about 1 to 4 days, the duct can no longer be seen on the echocardiogram. The ductus venosus we talked about, that is, it ceases to function the moment your umbilical venous flow is clamped and cut, right? So it ceases to function soon after birth, but the ductus venosus can still be seen up to about a week of age. And then it kind of disappears once again. In about a couple of weeks, it fibroses and anatomically also disappears and becomes a ligamentum venosum. So in summary, fetal circulation and neonatal circulation are quite different. So in the fetus, you have the placenta, you have ductus venosus with flow, you have the foramen ovale with right to left flow, and the ductus arteriosus, again, with flow from the pulmonary artery to the descending aorta. There is admixture at various levels. The desaturated blood and the less desaturated blood are mixing at multiple levels. But postnatally, what happens is the placenta is completely removed from the equation. Ductus venosus blood flow ceases or stops because of a drop in pulmonary vascular resistance. There is increased pulmonary blood flow, increased pulmonary venous return, increased left atrial pressure. The foramen ovale also closes shut. So there is no longer right to left flow shunt across the foramen ovale. If anything, there may be a left to right shunt. Again, because the PVR has dropped and the systemic vascular resistance is now high without the placenta, even the ductus arteriosus, even if present, will probably shunt left to right. Now, eventually, both the foramen ovale and ductus arteriosus will probably close. In a term baby, the ductus arteriosus almost always closes. The foramen ovale may remain small and patent in about 30% of adults without giving any problems. So this is how fetal circulation varies from neonatal circulation, where in the neonate, deoxygenated blood is completely separated from the oxygenated blood, and there is no admixture of blood. The two circulations are separate and they run in series. So I hope that this has clarified your understanding of fetal cardiac, development, physiology, and fetal circulation, and the transition of the fetus into postnatal life. Until we meet again, I'll say bye. And if you have any questions, please feel free to share them. Okay, bye-bye.